All right. So thank you guys so much for allowing me to come and talk today. Thank you for IREP for inviting me. So I'm going to talk today a little bit about some of our research addressing infectious disease biology and really trying to move into precision medicine of infectious disease and critical care medicine. So my lab is located in Grand Rapids, Michigan. If any of you guys aren't familiar with Grand Rapids, Beer City, USA, we have something like 100 breweries within the city. Our lab is located in the Grand Rapids Research Center, which is smack dab in the middle of a city environment. So, here's a research building. This is essentially a biotech hub. We've got a new clinical building moving in here. There's a brewery across the street. So it's a pretty cool lab to be able to work in. So before I get started, I just do want one small conflict of interest that I do have, which is that I worked for AbbVie Incorporated over the summer on a sabbatical, receiving hourly pay. None of that work is related to anything I'm going to show today. So really, those of you guys that know me, I know there's a few people on here that do, I'm what normally people refer to as an ultimate collaborator. And before I really present anything, I think what's so important to do is really make it clear that so much of this work is a team of many researchers, including those at IREP and many other places, Hudson Alpha, quite a few different colleges and universities, and really our clinical partner at Spectrum Health, where we really obtain all of our clinical samples that you'll see presented here. It really is a team effort. So before I get started, there really are 4 things you need to know about me as a person. One, a lot of my personality and reflection on science is driven by the fact that I've been studying rare diseases since I was in high school. So this is actually me in high school. And I was digging through stuff the other day, and I actually found this. This is my first Western blot I ever ran when I was a junior in high school, doing subcellular localizations with sucrose gradients, which thank goodness we don't have to do those anymore. Way back when, looking at proteins like the Menkes protein, trying to understand how variants in Menkes gives rise to rare diseases. The other thing you need to know about me is that oftentimes, while I've trained a lot as in genetics, so much of my personality is built on my original training as a physiologist, and oftentimes I think more like a physiologist. And in particular, the class that always sticks with me through my whole life was a course I took in stress physiology as an undergrad, where every day when we came into the class, we did what they call the cold pressor test. Right? You stick your hand into a bucket of ice and you measure your blood pressure. And what that always taught me is that responses are variable. Right? Everybody that does that is going to have a different response in blood pressure that varies. Not just that, but if you measure your blood pressure over multiple days, it's going to be quite variable, right? So one day you may come in, your blood pressure spikes up. And that really, trains you into thinking about how, even though we are who we are, so much of our response to biology is variable based on how we are that day. And so there's a huge component of the environment in everything we do. Third, understanding the immune system is quite personal. So, about 5 years ago, at the age of 30, while I was still down at Hudson Alpha, I really started waking up with sore joints, dry mouth, itchy eyes, had some abnormal spots on my skin, really started making me think maybe something is up. It's not terrible, but it was when I got my genome and my genetics information back that I realized, so much of my genome started suggesting I had quite a high risk of various autoimmune pathologies, including that of Sjogren's syndrome, which really matches up to this list of phenotypes. And so like any normal person, I go to my doctor, I say, you need to order a panel of autoantibodies to tell me, is this real or is this just genetic risk that isn't true and manifest? And of course, the doctor pushes back and says, you don't have any joint dysmorphology. Why would we waste the time and effort to run it? And so I fought back and said, no, please run it. I really am concerned and I want to know. So finally, the doctor caved, ran it. And sure enough, they found autoantibodies that I was positive for. And it really started telling me, so much about genetics and how we use it and understanding things. It's really not primed yet. for understanding immune biology and so much of that. And we really need to start moving into that direction more and more in precision medicine. The fourth thing is that my lab really in the last 3 years upon coming to Michigan State has been really focused on how genetics and the environment interact. Everything from how chemicals regulate organ biology and genetics to what you'll see presented today on the immune system. So as I mentioned, my lab is in Grand Rapids, Michigan. And our building is located right off of this actual image. Right over here is the Grand Rapids Research Center. And if we go one block up, essentially is what we call the medical mile. And this is a ecosystem of academic, clinical, and more actually commercial entities that have really all partnered up into applying medicine and research together in an integrated fashion. And so this includes a medical school at Michigan State, a pharmacy school from Ferris State, a hospital system, which has a cancer center, a children's hospital, beautiful brand new DeVos Children's Hospital building, an adult hospital system, a cardiac care center, the Myers Heart Center, outpatient testing. We've got additional academic partners in the undergraduate medical health training space at Grand Valley State. We have a massive community college integrated in. And then on top of that, we have one of the largest non-for-profit research institutes in the Van Andel Institute, which sits on a very large endowment allowing for advanced genomic research. And so we have all of this within a square mile, which is such a cool ecosystem to be in this city-friendly space. And the cool thing about Grand Rapids too is that it's a pretty cheap city. So I actually live a block away from all of this, meaning that I can really be immersed into this healthcare sector and walk a block and be in the middle of all of it. And so when I got here 3 years ago into the medical mile in Michigan State, I started setting up collaborations and meeting with people. And really it was these 5 people that really helped morph so much of the research you're going to see presented today. Clinicians focused in medical genetics and ICU care, immunobiology, translational research, and then one of my really good collaborators, Ben Chen, who is a really, really good big data scientist who can do some really advanced stuff in computing that, while I can dabble in computing, I can't touch what their group can do. And we started strategizing what are the areas of genomics that could be applied into this ecosystem in Grand Rapids here, and especially focused in precision medicine. And we identified 3 major areas. In the NICU, applying rapid whole genome sequencing, which we've quite well done over the last 3 years, partnering with Rady Children's Hospital. Looking at genetics, and those of you guys that know me, especially from my time down at Hudson Alpha, know that I classically was a variant analysis guy, focusing on variants of uncertain significance. We've kept that workflow running and going in the lab. But the third area in PICU, the pediatric ICU and the adult ICU, really we wanted to start understanding what's happening in each patient. It's such a ripe area to try to apply and understand precision medicine. So those of you guys that are familiar or unfamiliar with the ICU environment, right? When you walk into an ICU room, you see something like this, where you have so many gadgets and gizmos and devices that are all used for various aspects of medical care. And that's because this environment and this ecosystem, you don't know what the patient's going to be like. You have such a broad array of trauma, organ failure, all the way to infections. And those infections are quite diverse, as you'll see presented in the rest of the slides. So it's such an environment that you really don't know what you're going to have with a patient, but yet you have to try to figure out and identify what's going on in a very short window. And so when I think about precision medicine, to me, this is the entity that my eyes jump to and say, this is where we should be applying precision medicine. If we can't do it here, we really can't do it anywhere else. And so a lot of this project really morphed into really trying to understand how the immune system responds in individuals to various infections. So as you'll see at the end, looking at something like COVID-19 and how these infections give signatures, but more so how these signatures are unique across different patients. And this has required us to think about the bioinformatics in a very different light. right? We've got a mixed population. And while many people and many groups really focus on building cohort comparisons, right? Taking the patients that are normal relative to those patients with a pathology, patients with a pathology relative to those that might be lethal, comparing lethal pathologies to other lethal pathologies, and creating genes and signatures that tell us about that pathology. We really have been more interested in pairwise comparisons of saying, how does each individual similar and different from each other that really starts informing at the patient level insight of the immune response. And so when we were starting to develop this project, we start off saying, well, what do we want to get? What type of information? And you'll see we decided on several, but really the staple of the program we're trying to create focuses around blood collection and working with the nurses in the hospital. What is the easiest platform for us to potentially collect in? It was really decided to use the PAX-gene tube system. Those of you that aren't familiar with PAX-gene tubes, right? Blood goes into the tube, you invert it, and it stabilizes RNA. And then once it's stabilized, it can sit in the freezer. It's really amenable to the clinicians, especially in an ICU environment where it is about patient care, not about getting samples, right? It is hard for clinicians and nursing staff and our clinical collection staff to get into these environments and do very detailed, laborious prep procedures. So the PAX-gene tube is very flexible for them, which then can be used for follow-up RNA isolation. So when you think about a PAX-gene tube, right, what is in this RNA? And we, of course, our original goal was to focus on the immune cells and the blood cell components. But as you're going to see, we started realizing there's so much more in it. That of the immune repertoire, that of bacterial RNA, viral RNA, potential cell-free RNA, and damage markers, all of which we can try to figure out information from it. And so with that, you know, as a bioinformatics lab, we started taking our bioinformatics side and applying strategies of taking that RNA-seq data. After it comes off, we do total RNA-seq, normally pretty long reads and very high depth. So right now, our standard RNA-seq is paired end of 150 with about 150 million reads generated per individual. We know we can really dampen down those numbers, but in the discovery phase, we really want to be able to capture as much information as we can. And so in this workflow, we're able to capture your typical gene signatures, which we can take through pathway clustering. We can take them through cell deconvolution analysis. We can take and look at isoform biology and isoform switching, looking at damage biomarkers, especially using our cell-free annotations, getting more and more into looking at variants and understanding how variants influence the RNA, and then things like the immune repertoire and various signatures from foreign RNA. So the original design, we went after 3 groups, and we have very small sample sizes, right? We're a new startup research project, so we don't really have huge sample numbers, but our goal was to try to apply these strategies and really ask questions, what can we see from the RNA-seq in various pathologies? And so the first group I'll show you guys is focusing on infant cases, so those of 0 to 7 months of age, with respiratory syndactyl virus, RSV. The next cohort looks at multiple organ dysfunction syndrome in a group of 27 very heterogeneous cases of medicine. And then as COVID started popping up, we really made a switch and a shift into applying these tools into COVID biology with 36 COVID samples through our initial workflow. We've now actually collected, I think the last time I heard was 400 samples already that are sitting in the freezer. We just secured some funding to do some additional RNA-seq. And we're hoping that we're really ramping up our collection procedure with that funding as well to ramp up our numbers in COVID sample collections. So like I mentioned in our analysis, many groups focus on taking, for example, a principal component analysis and trying to understand what's the difference between the control group and the patient with whatever phenotype we're looking for. And you'll see we actually do multiple time point analysis in these patients as well. We're getting blood collections, not just at the initial admission into the ICU, but we have follow-up transcriptomes as well on them. Most people are really interested in the stratification of groups and understanding what is driving that. our interest really has become trying to understand why and how each two dots differ and are similar to each other. So we start off with these RSV cases kind of as our starting point, right? It's pretty easy to take the data and separate controls to infections. One of the areas we commonly get hit on quite a bit is why are we focusing on the blood if this is a lung infection? And what we clearly can show is that when you look at the blood of these patients, they show similar responses, right? We see cytokine signatures, we see immune system stimulation happening, and we know that there are more systemic alterations in the blood that we can note. And so these types of analyses are fairly easy, fairly straightforward. Compare one group to another, account for co-modifiers of pathology. What starts getting more challenging is, again, trying to move to the patient level insight. And the easiest way to start doing this is to just simply take your set of genes. For example, I could take a set of 5 genes involved in severe sepsis and say, what is each patient's level of response at that 5 set of genes? Doing the same thing for something like cytokines, where we know the cytokines that go down, we know the cytokines that go up in infection, and we can essentially see that. But what you're going to see is that there's a huge stratification with the patients. So yes, there is a difference in these cytokines that go up in response to RSV and the cytokine signature, but it's not the same in every patient. So we really shouldn't just stop at saying these cytokines are up and that's how the biology works. We really need to think about how each patient's total burden is impacting. And you'll see, especially when we get to the COVID data, it really is impactful to think that way sometimes. What became incredibly enlightening in these RSV cases is when we look at outlier genes in each sample. And so what we do is we take the whole entire cohort and we convert all of the values, the transcript per million values of RNA-seq into z-scores across that gene, across all samples, and essentially take any gene that has a z-score greater than 4, and then we do gene ontology enrichment from that set. And that essentially tells us, are the genes that we're finding in each patient elevated, are they interrelated? And when you start looking at that, it starts telling you so much about these single patient level pathology. And what I want you to really notice is a lot of these patients, we started mapping genetic signatures of rare diseases. And we do know that these RSV cases, they're notorious for being reoccurrence in hospital systems. So we are really seeing what looks like these individual type of pathologies happening. And it really reflects on, we need to be thinking about the patient level insight and how we can use this data moving forward. And so that takes us into the second cohort, which again, I told you is much more heterogeneous in base. Way more diversity of the phenotypes. So multiple organ dysfunction syndrome is a set of defined standards of having 2 organs that are in dysfunction, right? Pretty broad general term. And it could be everywhere from brain swelling to alterations in the heart, lung dysfunction like ARDS, gut inflammation and gut distress, kidney failure, cardiovascular instability, all of these things. And really, you think about these patients, they're going to be very diverse. So I looked at this and said, this is the ideal population to see, can we really use RNA-seq to do precision medicine, right? Awesome population base. Not just that, but many of these patients were also on what's called ECMO, which just tells you the severity of these patients' organ failures, that they essentially have this bypass machine that the blood is being filtered and oxygenated and pumped by outside system. And for these patients, we were able to get sample collections before the ECMO was administered, and then after the ECMO administration as well, which has been interesting to also see how ECMO modulates RNA, creates a huge amount of challenge when you think about the clinical side of these patients are being kept alive by outside support, blood donations. It is a very challenging phenotype to try to grasp. And so it really pushes our test limits in precision medicine. So as I mentioned, groups like Bin Chen that we've been working with are phenomenal big data lab that can really take these gene signatures and we can morph out and figure out genes that really are likely biomarkers of severity and really understanding what those are. We know a huge amount of the severity outcomes, especially the need for ECMO, kind of has to do with what's called netosis. neutrophil extracellular traps. We know how devastating these things are to pathology, especially in COVID, and how they wreak massive organ dysfunction. It really is a huge portion of the COVID biology that happens in patients. Our group was much more interested in the precision medicine side, though. So how are each patient unique and what's driving these signatures? And so when you look at this cohort, right, this is normally a bioinformatician's nightmare. You look at the ages, everywhere from a few months to 17 years. You look at the races and ethnicities are all over the place, multiple sexes. And then you start looking at the etiology and the clinical notes and realize this is everywhere: septic shock, pneumonia, acute kidney injury, carbon monoxide toxicity. It doesn't seem like a good group that you would want to study unless you think about how we use this data to look at an individual. And that's really what we did. And I want to be completely honest, our group was blinded to all of those clinical notes when we did this analysis. And this is what made these findings and this work so awesome in my mind, is that all we had was a sample number, right? So was it sample 1, 2, 3? Were they ECMO mods or sedation control? What day sample was that collected from? So most of these patients had 3 sample collections: at day 0 when they first were admitted to the PICU, at day 3, and then at day 8. And then we knew sex. And we actually started telling the clinical, not even to tell us sex, because we can determine that ourselves, right? We just look for the presence of Y chromosome genes in the RNA, and it's very easy to sex the samples. So it's one more thing that we don't have to fight with the IRB for them to give us in the ultimate return. And with that, we essentially started going and trying to tell precision medicine stories about each patient, what makes them unique, what we're picking up, right? Something that I still am kind of amazed that I decided to do, because it really is a high-risk research project. And so where we started off was looking at things like gene panels. Just like you saw with RSV and the data we had there, we knew that gene panels could tell us something about patient outliers on pathology. And so, for example, if we take the work of Hector Wong, who has been in the field of sepsis severity, phenotyping, and genetics for over a decade, and really is the world's leader in this field, right? He has put forth multiple genes that drive severity. And we could go to our cohort and say those genes, where are they activated, right? These 5 different persevere genes. And we know 2 patients are clear outliers. And these 2 patients are going to come up more and more, especially patient 24, where IREP really was able to help us figure out what was going on in that patient. We could look at other genes. So for example, we could look at cytokine signatures. And again, you see patient 8 kind of as an outlier. And we could start going to the clinic and saying, yeah, patient 8 is weird. Something clearly is going on. And sure enough, when you do that and they start feeding you back the clinical information after we did the bioinformatics, that's when we start realizing this is a patient with some severe biology. It's a patient that died in the PICU or died shortly after leaving the PICU, has seizure biology, genetic variants. They had a metapneumonia infection. So it's quite complex biology going on with very severe case. And this was actually the only patient in the cohort that actually passed away. And sure enough, we could see already saying the severity genes are high. These cytokine gene signatures are really high. We could look at other things. We could start looking at the immune cells, right? And doing immune cell deconvolution and focusing on those genes. And you can start seeing things like patient 11 all of a sudden popped out at time point 0. And sure enough, this is a case of Streptococcus pyogenes infection. That tells us, yeah, this makes sense. Neutrophils are activating to a strep infection. That's very useful information to know, and would be something that could start guiding me down that realm of saying patient 11 is having that type of response from the RNA-seq in the blood. You're going to see a lot of this. We knew that there was a lot of infection biology. You go to the PICU, you go to the ICU, a lot of it is infectious disease biology. So we really started asking questions: how much RNA is present from these RNA-seq that map to other species? And when we do this, we actually use about 30,000 level of annotations. That's about a terabyte worth of transcriptomes that we filter every patient through. And we can normalize those reads to how many reads we map to the human. And so you can see for every 1,000,000 reads in the human we map, we're mapping much fewer reads to bacteria and viruses. If we zoom in on those, you can still see it's still in the thousands. And so when we get very high density RNA-seq done, it really starts becoming informative on bacterium viral signatures. And when we're doing this is really one of the challenges in looking at these signals is so many of the times, there's a lot of RNA and RNA-seq that we don't really know where it comes from. It could come from the reagent prep side. It could come from just random here or there. So to try to account for those types of signatures, we really focus on outlier mapping, right? We're really interested in when is there a sample that is divergent from the rest of the cohort. And so if we look at something like those mappings for Staphylococcus aureus or Streptococcus pyogenes infections, you'll see very strong outliers in the data set. And sure enough, if we look at something like patient 7, which at time point 8 showed a spike in staph infection, this was a clinically confirmed hospital-acquired staph infection. And we're seeing that at time point 8 in that patient. If we look, for example, at the strep infections, we can see patient 11 was a clinically confirmed Streptococcus pyogenes infection. And sure enough, we see this very strong as an outlier mapping. Same thing with patient 5. Clinical group A, beta hemolytic Streptococcus, and we're seeing reads across Streptococcus. If we look at, for example, this back to patient 8, which I told you was this patient that passed away, there was very clear evidence that this patient had some type of infection, and the clinical staff could never figure out what it was. And when we started looking at the outlier mapping for this patient, especially at time point 8 when they're at the height of their infection, sure enough, we started seeing outlier mappings for bacteria like this Cloacobacillus porcorum, and I apologize if I slaughter that pronunciation. But what's interesting is this is a anaerobic infection, right? This is not something that would culture easily in a culture setting, so very hard to confirm. There are reports that this anaerobic bug can be drive bacteremia. The clearance of this requires a very different anaerobic use or a very different antibiotic combination than what is typically used within a clinical setting. So it starts adding up and making sense. And I think this really highlights one of the things we're discovering, and I think other people are doing this at the DNA level as well. For example, Rady Children's has a wonderful program looking at this, is that the role of anaerobic infections is way higher than we've ever really appreciated, especially as co-modulators of infection outcomes. These anaerobic bugs don't culture, and so we oftentimes don't really know that they're there. And genomics and transcriptomics is a really awesome potential to be able to see these bugs for the first time. We can also look at things like viruses. So for example, the herpes viruses. We know that several of these patients had herpes viruses. We can see, for example, in patient 20, a high level of beta herpes virus 7. In patient 9 and 24, a high level of Epstein-Barr virus. And sure enough, these are very complex cases, but patients like 24 had an EBV detected PCR. And so we really are hitting on these. And I really want to leave you with that lingering thought of, why haven't we been using RNA-seq and these types of platforms to detect infections? It could be done way quicker than culturing plates. Plus, you're going to detect things at limits that were undetectable. And especially looking at more outlier things. One of the things that really threw us for a loop was this patient 6. When we did this mapping, we started seeing measles RNA present in this patient. And being a little bit of micro background, right? I know measles is something I have to go to the clinical team and tell them. I have to report that to them when I notice this. And it's not just one measurement, but both at time point 0 and at time point 3 of our collection procedure, day 3 in the PICU, we see measles RNA. And it drops out at day 8. So, I took this case. There's really no presentation of measles in this. And so one of the things that I did was went back to the clinicians and said, what is going on? It doesn't look like there's measles in this case. The doctors kind of put their heads together, started thinking, we have no idea what's going on. All of a sudden, one of the doctors is like, well, if they received the measles vaccine, that's a live attenuated vaccine. That could be the RNA. They then went back to the medical records and looked, and sure enough, 3 days before they showed up in the PICU, they had the measles vaccine. And so we're seeing the signatures of that. And so when you think about what happens when you get a vaccine, which is stimulating your immune response, and you get unlucky and you get a Pseudomonas infection at the same time, right? Is it that double combination? The vaccine itself is pretty safe, but when you put that stimulation of the immune system on top of a stimulation of another infection, you start realizing the immune system can be overtaxed and stimulated. We've also been doing a lot of work looking at organ damage biomarkers and looking at these cell-free signatures in the RNA. This was actually driven by the detection of albumin, which we realized in a few of the patients, we saw RNA for albumin. Albumin is only made by liver and the hepatocytes. So right away it becomes, this doesn't make sense. Well, sure enough, if you look at the patients that we mapped albumin in, we could see that the reads actually mapped over the entire transcript. We're seeing hundreds of reads that map to albumin, highly specific. It's not a misalignment, not a misannotation. If you look at the samples that had a spike in albumin, they were both at day 0 when they came in. And sure enough, by day 3 in those patients, they started spiking AST, which is a clinical biomarker for liver damage. And so it actually suggests that something like the presence of albumin in the blood is a way earlier prognostic indicator for damage to the liver. And I want to highlight, these RNAs are clearing out really fast, right? By the time you get to day 3, they've completely cleared out of the system. So we've been trying to think about how we apply this to other organs, cell types, focusing on cell-specific genes, cell type specific genes that we can then go into the patients and look at additive properties of multiple genes in each tissue type. One of them that we put a lot of focus on is actually the kidney, where we've combined both our RNA-seq data with proteinuria and tubule damage biomarkers in the urine. And creatinine, which interestingly, creatinine showed a really interesting story with streptococcus infections, that if you have a strep infection, it elevates creatinine in the urine. But by combining these, we're able to actually figure out patients that truly have damage in the kidney. And interestingly, it gave some insights that there's quite diverse biology happening with our biomarkers. And there's some isoform switches. And it has to do with whether or not it's virally activated in the blood, and then you end up with proteinuria. And so of course you pick up the biomarker if it's elevated in the blood and then drive something, as opposed to if it's truly elevated in the tubules and that the spike is driven by the tubules without proteinuria, in which case then we could segregate the marker genes that are specific to the tubule cells and actually show and confirm that they are actually present in these 2 samples and not any of the other samples. And so it really has started linking and showing this is a true phenomenon we're mapping with the patch gene tubes. The really cool stuff, and what brings us into our IREP work, is focusing on multiple time point comparisons. And this is really trying to take our day 0 versus day 3 and 8 and understanding the complex biology happening. For example, this patient with alveolar rhabdosarcoma, the genes that you see elevated when they first showed up in the hospital are highly associated with herpes-induced rhabdosarcoma. And they, again, had a herpes virus infection as well. Some of the more diverse cases, for example, this carbon monoxide poisoning case, we could use the RNA-seq, understand the immune stimulation happening. We also paired this with some lipidomics in the patient, really because of the phenotype in this patient, where it wasn't just a carbon monoxide poisoning. They also had a viral infection that was detected, and they ended up developing Parkinsonian's tremor. And using this data, we were kind of able to, what we think in an N of 1 strategy, try to piece together what might be happening to drive that Parkinsonian's-like phenotype. This is really challenging. Anybody that does N of 1 genomic science knows N of 1 science is really hard. So I think this is a real challenge of understanding, is this true? The combination of phenotypes in this patient, I'm going to guess you probably have to go to 100 hospitals before you ever find, over a course of several years, find a patient that had the same exact set of environmental regulations happening. So really it starts making you realize and think that we need to be thinking about infectious diseases as precision medicine. And the patient that kind of highlights the need for this is patient 24. And this patient 24, when we looked at multiple time point comparisons, we actually started getting what we thought were syndromic biology signatures coming out. It's really the only patient we see shifting in this fashion for genes. And this is a patient with septic shock. and a very advanced immune system overregulation. And so if you look at this patient's time course, about 10 days before they showed up at Spectrum Health, they actually went to the doctor's office. They diagnosed him with a bacterial infection, gave him amoxicillin, sent him home. And then they went into slow organ failure. And within 8 days, they were in the ER. Within 2 more days, they were administered to the PICU, put on life support. And you can see the phenotypes down here really start giving you that signature of sepsis. For example, hyperferritinemia as a spike. And this really led the doctors into looking at things like HLH, which HLH is oftentimes genomic in nature. So they ordered a rapid whole exome to be done as well. Thank goodness they used a really good strategy of treatment, which is a combination of anakinra and dexamethasone. We're able to save the patient's life. And we were able to get multiple time points of RNA-seq. So we could try to understand what's happening throughout this time course. So again, this is a patient that had an Epstein-Barr virus infection. And we can see the RNA signatures from Epstein-Barr virus in that patient at day 0. This is actually what first led us to realize we could map bacterial infections really well in these samples, was we looked for this. Not just that, but the genomics returned variants in this gene, RNA-seq2b. And this is a gene that is a autosomal recessive gene for a syndrome called Aicardi-Gouttières syndrome, which is a very devastating phenotype. The kids very rarely live past a few years old with the phenotype. And this is a kid that was 16 years old and totally healthy before this. So it didn't make sense. And when we looked at the report, one of the variants, which was this valine to leucine change, using our VUS characterization tools, we knew this wasn't a bad variant. The other variant was a splice variant. So we started digging into the RNA-seq to ask questions, does the splice variant potentially cause a termination of the protein? And we actually realized it was really hard to find the mom's variant in the RNA. And so we started doing allelic imbalance analysis to look at the RNA being made from mom versus dad's inherited allele and realize there's a massive skew in the imbalance. And mom's RNA is being suppressed, but at the height of the Epstein-Barr virus, it really wasn't suppressed at the same level. And so this concept is what's known as nonsense mediated decay. And interestingly, when we look at day 0 in that patient, we could pick up mom's variant and confirm the presence of a frameshift mutation. And that frameshift mutation is going to completely cause what we know to be a dominant negative protein in RNA-seq2b. So you've got a patient that the virus is turning on this pathology. More so, NMD is this pathway that normally is used by a cell to regulate degradation products. So if you've got a missense variant or a nonsense variant or frameshift variant, you don't want to make that protein because it potentially could make half of a protein that's very dominant negative in nature. And so the cells have mechanisms to get rid of these things. Well, it turns out that these processes also inhibit viruses themselves and inhibit the viral RNA because viruses tend to have very long 3' UTRs, which is how NMD works. So viruses come up with mechanisms to inhibit this. When they inhibit it, that actually turns on a handful of innate immune responses, which are good and stimulate and say, hey, there's an infection. It allows the virus to replicate. And then the virus also develops mechanisms to then suppress this innate immune activation. So it's a double whammy of how viruses evolve to become efficient in cells. And when this happens, that essentially is turning off nonsense-mediated decay in a process we've coined viral-induced genetics. And it's kind of like an antivirus on a computer. You essentially have your computer, the antivirus is normally working. The thing a virus normally does is first try to target the antivirus. And when it takes it down, oftentimes that antivirus is actually keeping a whole bunch of corrupted files on your computer from going haywire. And essentially these corrupted files then drive a huge amount of the outcomes from a virus when your computer goes down. And so when you really think about it, if you have one of these variants, a nonsense or frameshift, your cells are really good at hiding it and keeping sometimes, especially in recessive disorders, from that working. All of a sudden the virus steps in and regulates it. And with this, we really knew that this patient had some type of major immune complication. Turned out that the patient had actually been in and out of the ER I think it was something like 12 times over the next 2 years. And so we decided to do a follow-up analysis with the patient to see, is their immune system still really dysfunctional? And sure enough, it was. It was still really heightened. Seemed like NMDA, nonsense-mediated decay, was still really screwed up. There's still this massive interferon signal activation. And so we reached out to IREP and actually said, this might be a really good patient to try to do immune system profiling with, using the immune repertoire analysis. And sure enough, when we got the data back from IREP, right, you can see that there is still some major activated immune component going on here with an overlapping signal for something in the immune system. And so it really tells us this patient does still have an altered immune system. And that actually was enough guidance to really start making sure there's standard meetings with the immunology team for this. We still don't know which came first. Was the immune system profile a driver of that original EBV infection? And this really is driving us to start thinking about how we get samples before infections happen, which we're working on really hard in the hospital system. What's really interesting about this patient, this time course should sound somewhat familiar actually, because this is actually the time course of COVID. And so when we figured out the biology of this, we really made a pivot and said, why don't we start trying to explore why there are severity outcomes in those COVID patients that move into the same organ failure? And sure enough, we actually were kicking and screaming at this time when COVID started breaking out, saying these patients need to go on dexamethasone. We know that dexamethasone works really well in these patients. And you go talk to most pediatric doctors that work with HLH, they know that dexamethasone is the drug of choice. It just happened to take a very long time before dexamethasone finally kicked in as being one of the main drugs of choice now for using to treat these multiple organ failure patients in COVID. So with that, we really launched an entire year of COVID research projects, trying to explore the virus and how proteins work. I'm not going to go too much into this. We published this in JBC just recently. But I will hype up, this really is one of the cool things about having a large network of universities that we work with, including Hudson Alpha and the CODE program there, where we can mobilize a network of undergrads to understand all of the proteins of the virus and how they work, which we actually beat out every other group that was doing protein dynamicomes and protein-based evolutionary mapping, and published this first paper in JBC before most other papers actually got a paper published even into bioRxiv. And we launched this VISTED database. You can go and Google VISTED and get resources for understanding each of the proteins. And we wanted to start exploring which of these proteins may regulate NMD in the cells and the genetic mechanisms to it. And realize that there's one protein that's kind of the central anchor of this in COVID, and it's the nucleocapsid protein that regulates all the components of nonsense-mediated decay. We also then asked detailed questions on how exactly does nucleocapsid work. And a lot of this is ongoing research projects still in the lab, so I can't really say too much about it yet until it's all done. We've been looking at that innate immune system regulation as well of this NMD regulation pathway. And we know proteins like the papain-like protease and ORF8 are key regulators of this in the cells. We also are really interested in the genetic outcomes, and ORF8 is one that I'm fascinated by because the proteins that it interacts with have a high platform of known genetic pathologies connected to them. And so if we're rederiving the function of a lot of these proteins, we can think about how ORF8 is really a driver of the phenotypes of COVID. But more so, we really wanted to make that pivot into the RNA-seq and understanding what's happening in these patients. And this is still ongoing work, so I'm just going to show a little bit of it. Our cohort had 11 patients with lethality, 36 total COVID patients and 15 controls. Quite a diverse population for our initial run. We had multiple time points on these patients. And sure enough, we can see really strong, robust signals in the blood of the patients, which we can do gene analysis to say, here are groups of genes at work. From those groups of genes, this is actually one of the things I want to really highlight. When you look at a group of genes in patients, right, again, we can see how some patients are much higher than others, clearly all elevated relative to the controls in the black dots. What we're actually looking here is a two-dimensional analysis of the summed transcript per millions of expression, and then the summed Z-score of all the genes. And so as you skew up, it's essentially a 1-to-1, right? You have a lot of genes turning up that all turn up at high levels. As they skew off to the branch here, we actually see there's a subset of patients that only activate a subset of the severity influenza linked genes, suggesting there are subsets of COVID patients. Other types of gene sets, we don't see that so much in. Where we really see a lot of this subset biology happening is that of interferon responses, right? If you go look in the literature, it's kind of a confusing thing of normally we talk about how COVID patients are interferon suppressed. They have a suppression of interferon activation, but it all really matters when you look at the patients. And what we're actually seeing is well, most of the patients truly are interferon suppressed, suggesting they're having a dampening of interferon responses. There's a subset again that are overactivating and really driving this. We looked at other indicators and building correlations, for example, the simplified acute physiology scores, or the SAPS-2, which bring together a lot of clinical parameters. And then we can look and say what set of genes correlate with those severity outcomes. And we know that a lot of this has to do with overactive immune system, but we then realize there's a subset of patients that actually is more divergent. And if we look at the gene signatures of this subset of patients, this is actually autoimmune pathologies. And so we're starting to see that there are some patients that overstimulate the immune system, some patients that are having autoimmune pathologies happening. We also know there's a heavy skew in the cell deconvolution, a heavy reliance on activation of neutrophils, some activation of T cell CD4 memory responses, and then a massive downplay of CD4 memory resting and the B cell memory cells in many of the COVID severe patients. And lethality has a little bit of a skew as well in this. What really got fascinating was when we started getting to our infection mapping, that we saw quite a bit of Pseudomonas infections, not just the Pseudomonas in general in patients, but we also could pick up their phages as well. We could pick up a handful of viruses. For example, patients that have activation of EBV, that we know that this virus can reactivate the EBV if it's in the right position. And this reactivation essentially drives a portion of pathology for that subset. Interestingly, we see the same thing happen with shingles, some viruses like torquatinovirus, which I actually was not very familiar with. And I go ask all my med students all the time, do you know torquatinovirus? And nobody hears about this, but it's a pretty common normal virus that doesn't really have any known connection to phenotype outside of those immunosuppressed patients. And so it's suggesting that these patients have a subset of activation when they activate that way. One of the really cool things is we can actually, by sequencing such high depth in these samples, we can pick up COVID in the blood in a handful of samples. And so we've been doing work on genotyping from the RNA-seq as well for COVID. We've been doing some immune repertoire analysis with this data, and we know that there really is not much of a difference between lethality, which is in the red and orange, as opposed to patients that survive, on whether or not they're really activating the immune repertoire. But it really does seem to be activating pretty evenly, with later time point collections much higher than the initial ICU admission. And we've been doing some work trying to figure out, are there any immune profiles that correspond to lethality or various levels? The really cool thing has been in looking at some of the outlier pathologies and thinking about the outlier genes in patients and how they link to things like cardiovascular complications, nervous system complications, blood issues, and really trying to get to what a lot of other people are seeing is that there is a rare disease biology component in COVID severity outcomes. For example, mutations within genes in the interferon response pathways. And so we've been now actively working on trying to take RNA-seq through variant analysis pipelines, looking at things like the severity GWAS data sets, using RNA-seq to build insights on those, going into patients and actually looking at the genotype off the RNA-seq. So we find any variant that's in LD with the lead SNPs that has a presence within a transcript, and then we can go genotype from the RNA reads. But what this has really given us is our new ongoing project of trying to take these blood samples and link it to patient records now, really in this new precision medicine strategy, to look at all these signatures and then link it to a REDCap database of the clinical data, to start building more longitudinal biomarkers, especially for chronic pathologies of COVID. And then another aim that we don't have funded yet, but we're really trying to push hard. Maybe we can convince NIH reviewers it's worthwhile to do follow-up RNA-seq on some COVID patients. And so with that, I really want to thank you guys for joining me for this talk. I know it's a lot of data. Hopefully I've convinced you that there's a precision medicine component in infectious disease biology. So I think we're going to take some questions now.
All right, great. Yeah, so we do have some questions that have come in. For the whole blood collection in PAXgene, how many tubes do you typically collect?
So a lot of that depends on the age of the patient, which is a huge challenge. In an adult, for example, the COVID studies, we collect two 2 milliliter collections, and one tube is essentially more than enough to get 150 million read, 150 base pair paired end RNA-seq. Right, we have pretty much, we can collect as much blood as we need to from those patients. And then we biobank one of the 2 tubes, which is especially important as we talk about partnerships with IREP and others of what do we do as follow-ups if we discover something, just like those other patients. It'd really be nice if we can do detailed follow-up analyses when we find something with the RNA-seq data, especially because oftentimes we burn all the RNA for the first analysis. When we look at something like RSV patients, obviously these are infants, and so getting blood is very challenging, especially the younger and younger they get. And so we were really limited in the RNA on a few samples to sub 1 milliliter single PAXgene tube collections. So it's variable. And again, this is one of the challenges of IRB protocols, is figuring out what is allowable. As somebody who's not hardcore clinical and IRB minded, it really was an eye opener to think about, you really need to limit how much blood you're collecting from an infant. It makes a lot of sense, but the researcher in me always says, well, can't we get a little bit more? And then the humanist in you comes out and like, it really is, you have a very small collection. So.
Yeah, right. Yeah, and then I think you answered it with that. Just a second part of that question was just, can you extract and do the analysis from one tube? It sounds like you can.
Yeah.
Okay.
Almost always we're just doing this from a single PAXgene tube. And so it's nice when we can get a second tube to have that biobank. And the really nice thing about partnering with somebody like Spectrum Health is they have a phenomenal biobank. I didn't really mention much of that, but they essentially, when COVID started happening, we had our IRB approved to be collecting samples. I think within by end of March, early April, we were already collecting COVID samples and getting the sample. Then it was a challenge of coming up with the money to do the RNA-seq. So
nice. Now it seems that one of the main advantages of this approach is identifying comorbidities. How do you see this used going forward?
Can you repeat that question again?
Yeah, so one of the advantages of this approach is identifying comorbidities.
Yeah.
And how do you see using this information going forward?
Yeah, I think that is ultimately the challenge of precision medicine. And I think one of the things we started seeing in this is oftentimes there are comorbidities that the clinicians didn't even annotate. And when we went back, or how exactly the clinicians annotate it, it really is a challenge. If it's in the notes, that is not a simple data extraction to take the note details and bring in. So one of the things we actually saw a lot of was when we matched something, and I reached out and said, for example, some of our controls in COVID, and actually this has been one of the biggest challenges, is what are controls when we start doing these outlier mappings, because we see this control actually had an infection and the control has heart disease. We had controls with viral infections. And they're healthy individuals that walk around with a long-term virus or something. We start picking up these signatures. And so we take that stuff back to the doctors. And oftentimes these are things that are actually in the notes. And so it actually has been really useful in the comorbidity space to help elevate note documents when we're working with our clinical data miners.
Mm-hmm. Great. Let's see, the next one. Can signatures of organ failure be detected early enough for intervention?
So this is actually, This is I think that the quintessential question, not just to organ failure, but to everything, of how do we get this data fast enough? All of this that you saw presented here, what was in the biobanking samples, collecting it, and then running batches of RNA-seq. We're trying to think about how do you actually make this in a real-time procedure? And so one of the things we've been doing, right, tools like, for example, Nanopore, where a run starts giving real-time data instead of a 24-hour run of an RNA-seq, we can actually have 1 million reads. And we've been doing this with a few samples. We piloted this actually with the patient 24 that had that complex NMD-regulated biology. We split the sample in half, and we did half of it with a Nanopore platform. And sure enough, you can pick up within an hour 1 million reads and then put a floating bioinformatics workflow on top of that. And it's possible to start getting data. The challenge there is that Nanopore does not do some of the other mapping strategies because it's really limited to poly A. And so we don't see all of the same bacterial and viral signatures. It becomes much more susceptible to degradation of RNA. So our organ failure stuff, and the organ pathology biomarkers, degradation is a huge issue. And so when you're doing poly A, these RNAs that are just floating in the blood are going to degrade much quicker. And so we saw some of this. We saw a drop out of a lot of that signature. So I think it's a challenge, but a challenge that I think that the future is going to be able to figure out for precision medicine. And I say that with hope because we've done it for genomics. I mentioned at the beginning of the talk, we can ship out a sample to Rady Children's and have a genome returned in 2 days with trios. If we can do that for a genome, I assume we could do it for an RNA-seq run at some point. And maybe push that timeline down to 6-12 hours to data return, which is actually now outcompeting something like culturing in labs, starting to compete with PCR-based timelines. That's our ultimate hope. But I think it is the biggest challenge of moving this really into clinical utility. So I think that's a wonderful question.
Yeah. Yeah, that's great. And I think that covers like the next question really about about using RNA-seq as in clinical utility. But so could this type of investigative research be useful in determining the lethality of COVID infection, especially with new strains popping up?
Yeah, I think this is, so we just submitted this stuff for publication. I assume the reviewers are going to hit us on this. A couple of my co-authors really wanted us to try to figure this out. To be honest, our gene signatures don't give us much on lethality. When you look at those patients that are lethal versus not, the better stratifiers are things like age. And we actually started realizing a lot of it is these nested N of 1 stories. For example, one of the lethal patients had an activation of EBV, that double insult. We have these autoimmune pathologies that activate in some patients where we can actually see the gene signatures and the IREP data, or the immune repertoire data, corresponding really well to autoimmune pathology that drove lethality. But other patients are having, I didn't really talk a lot about it, are having these overactive netosis processes driving massive endotheliopathy complications and these organ failures with the cytokine profiles. I think what this really is we need to think about lethality not as a group, but precision medicine. And I think it's the hardest thing I've had convincing people, especially funders, who always are used to thinking about the patients that die are all similar. But I showed at the beginning of my talk, when you measure your blood pressure, when you stick it in a pot of ice every day, it's different. Some days your blood pressure may get to the point that you die. Some days, if I were to stick that in there, right, I might die because I have an infection, right? It's not one pathology, but thinking about more of the precision entity. And I think it's really what we're noticing with this data is it doesn't stratify and cluster well. And so these classical arguments of you need to elevate your sample size to find them, what that's really doing is enriching for a subset of individuals to find out what's happening to them. And that might account for 50%, 60%. We do a really bad job of doing metrics on how many people actually truly match to that biomarker. I am, again, this rare disease biologist in me is always focused on really understanding the multitude of responses that drive lethality.
Great. Yeah, thank you. I think that's all the questions. Jeremy, again, thank you so much for your talk. It was really informative. Thank you to everybody who attended. Everybody have a wonderful day. And again, thank you so much. Take care.
Thank you. Bye.